So today, we're going to dive into a molecule with a pretty bold claim, SH-1. It's a new compound from Sovereign Diagnostics, and its whole goal is to completely rewrite the story of hair loss. Let's get into it.
Right from the get-go, the source material sets this really powerful, almost philosophical tone with this quote. It's framing hair loss not just as some biological thing, but as losing a part of your personal story. And SH-1? Well, that's the act of defiance.
And for so many people, that's exactly what it feels like, right? The most common kind of hair loss, androgenetic alopecia, is usually seen as this unavoidable march, a slow, hormonal takeover where the follicle just gets smaller and smaller until it basically gives up.
Okay, so to really get the solution, we have to understand the problem. We have to understand the antagonist of our story. And in the world of hair loss, that antagonist has a name, dihydrotestosterone, or as most people know it, DHT.
So what exactly is this stuff? Well, DHT is a really potent hormone that's actually derived from testosterone. And for people who are genetically prone to hair loss, DHT is the main villain. It latches onto receptors in your hair follicles and kicks off this process called miniaturization, which is just a fancy word for shrinking.
The whole thing works like a quiet sabotage. DHT shows up at what the source calls the follicular gate. It successfully binds to the receptor there, and that single connection is the trigger. It tells the follicle to get smaller, weaker, with every single hair cycle, until its story just fades away.
But what if you could stop DHT right there, at the gate? This is where our protagonist, SH-1, enters the picture. It's being positioned not really as a cure, but as, well, the creators call it a sovereign agent of resistance.
So, SH-1 is engineered to be a non-steroidal antagonist. Now, those are really key words. Non-steroidal means it's not a hormone itself, which is super important, and antagonist means it's built to block something, in this case, DHT. It fights back, but not with brute force. The source calls its method elegant receptor-level defiance. All right, so let's get into the really cool part, the core mechanism, which the source calls the docking duel. This is all about how SH-1 is designed to literally outsmart the very receptor that DHT is trying to attack.
Now, what's so fascinating here is the difference in strategy. You see, conventional treatments like finasteride, they work by going after the enzyme that makes DHT in the first place, kind of like shutting down the factory. But that can sometimes lead to broader, systemic side effects. SH-1 does something totally different. It ignores the factory completely and goes straight for the target, the androgen receptor right on the hair follicle itself.
Now, to understand just how well this works, we need a quick science term, binding affinity, or Ki. It's actually a pretty simple concept. It's just a score that measures how tightly a molecule sticks to its target. And all you need to remember is this: the lower the Ki number, the stronger and more effective that bond is.
And check this out. Here are the numbers. DHT, the molecule we're trying to block, has a binding affinity of about 1.9 nanomolar. SH-1 is right there with it, clocking in at around 2.3 nanomolar. I mean, those numbers are incredibly close. It's like two people racing for the last best parking spot right in front of the receptor. It means SH-1 is a very competitive blocker.
So how does it win this duel? Well, it docks with amazing precision. It uses 3 hydrogen bonds to lock itself in place, and it creates this physical shield. But here is the absolute genius part. It binds, but it refuses to activate the receptor. It's like a key that fits the lock perfectly but won't turn, jamming the mechanism so the real DHT key can't get in and cause any damage.
Okay, so the theory sounds amazing, right? But does it actually work in practice? Let's move on to the preclinical evidence and see what happened in the lab.
And wow, look at these results. This really shows you what's possible. In the early trials on mouse models, using SH-1 led to a 50% increase in hair density and a 50% increase in the diameter of the hair follicles. That doesn't just mean more hair, it means thicker, stronger hair.
So what about safety? I mean, that's the other huge question, right? Well, the early toxicology reports look really promising. Because SH-1 is designed to work locally, right at the follicle, the data shows no systemic hormonal drift. That means it doesn't seem to wander off and mess with hormones in the rest of the body, avoiding some of the side effects that can be a concern with other treatments. The source sums it up perfectly. No compromise.
So, what's next on the horizon? Well, the project is now moving towards human trials. It's going to start with phase 1, testing a topical formulation. After that, they'll figure out the best dosages and eventually test it across all sorts of different people and hair types.
All right, let's circle back for a second to the philosophy behind this molecule, because according to the source material, this whole project is about something much, much deeper than just cosmetics.
They frame the goal here as reclamation, and the idea is that by bringing a hair follicle back to life, you're not just growing a single strand of hair, you're reactivating a piece of a personal story that was being erased. It's a really powerful way to think about the psychological weight of hair loss.
This quote just nails the whole idea. The people who made this don't see SH-1 as just another medication you pick up at the pharmacy. They see it as a tool for people to refuse a biological narrative that they just don't accept. It's an act of sovereignty over your own body.
And that leaves us with this final, really provocative thought. It takes us way beyond the science and asks a much bigger question about identity, about defiance, and the stories we tell about ourselves. If a single follicle is a story, what does it truly mean to refuse its erasure?